A very good morning to everyone and welcome to the webinar hosted by Additives. Today we will have the pleasure to discuss about an always present challenge, the heat stress, and in particular on how to adapt livestock production to climate change and the role of nutrition. Before jumping into the presentation of our speaker, let me first introduce myself and share the few housekeeping rules that will allow us smooth and clear communication during the webinar. My name is Ricardo Comunò. I'm Additives Country Head for Australia and New Zealand, and today I'm honored to be the moderator of the session. Now the two housekeeping rules. By default, every one of you is on mute. On the right side of your screen, you can see two sections of the question and the poll. Polls will be introduced during the presentation and you will be asked to participate at the right time. About the question, all of you are highly welcome to key in them at any moment during the presentation. They will be answered at the end in a specific session. Now, let me walk you through today's program. First to speak will be Professor Frank Danchet, who will be discussing and sharing update and new insight on the nutritional mitigation of heat stress in livestock. Professor Danchet is an internationally renowned animal scientist with more than 35 years of activities. He is currently Redmond Berry Distinguished Professor, Professor of Animal Growth and Development at the University of Leeds, and Chair of Agriculture at the University of Melbourne. His area of expertise is in growth physiology and nutrition and understanding the interaction between the animal and the animal's environment, with particular focus on the heat stress and its benefit on livestock. Before starting Professor Danchet's presentation, let's all now start answering the first of the four polls that we will be present today. Professor Danchet, for the moment, you can share your screen and be ready for the presentation. The polls will be updated online in few seconds. You will find them live with a red dot blinking on the poll section. Okay, now the first poll is live. All of you are welcome and ready to answer. Okay, thank you very much, everyone, for taking time and answering the first poll. Thank you very much, Professor Danchet. Now you can start. The stage is all yours.
Okay, thank you very much, Ricardo, for that introduction. And hopefully everyone can see my title slide. So today I'm going to talk about nutritional mitigation against heat stress in livestock, with a particular focus on pigs and on ruminants. So this will be the structure of my talk. An introduction, I'll talk about oxidative stress and heat stress, and then talk about a number of different strategies that we can look at to help protect us against heat stress, and then finally some conclusions. So why would we focus on heat stress? Well, I think this slide simply shows that there's been a dramatic increase in both the frequency and the magnitude of heat waves, which we're classifying as 3 or more days of high maximum and minimum temperatures. So these are becoming more frequent, and the duration is lasting a bit longer, and they're becoming more severe. This is just not a problem for animals, it's also a problem for humans. And I'd just like to point out that Heat actually kills more Australians than any natural disaster, and heat stroke is fatal in up to 80% of cases. So some of the things that I'm going to talk about today with respect to our livestock species is also critical for humans. And so we must also take care of people who are particularly susceptible, which includes the elderly and those that may be suffering from diabetes. And the link to diabetes is important because I'll talk about insulin sensitivity during my presentation. This is a slide that Jeremy Cottrell developed a number of years ago, and I'd like to use this because it is one way of showing the multitude of things that happen as the environmental temperature is increased. So as we increase environmental temperature, one of the normal adaptations is to reduce feed intake. It is to reduce activity, and in lactating animals to reduce lactation. All of these are aimed at reducing metabolic heat. Now, another reason I like to use this slide is because it identifies where we are looking at targeting some of our nutritional strategies. So when I talk about this decrease in metabolic heat, one of the strategies, betaine, that I'll talk about, is actually focused on reducing metabolic heat. If we step down to the next line, we see that one of the adaptations to heat stress is partitioning of blood flow away from the gastrointestinal tract to the periphery. Now, this aids in radiant heat loss. So what happens there is there's a dramatic increase in the amount of blood that's going to the periphery. There's a reduction in the blood that goes to the gastrointestinal tract. And one of the issues is that we're not getting the same amount of oxygen supply to the important visceral tissues and gastrointestinal tissues, and we can get hypoxia, oxidative damage, and ultimately an increase in gastrointestinal permeability, which can result in the influx of endotoxins and other contaminants. Another adaptation during heat stress is an increase in respiration rate, and this occurs to increase evaporative heat loss. So we see dramatic increases in the panting, and ultimately, if heat stress is too great, we'll see open mouth panting with heavy salivation. So we're focused on looking at trying to reduce this as well. Now, one of the consequences of increasing respiration rate is that we can result in blood alkalosis and an increase in maintenance energy. Again, we're looking at some of our strategies to reduce that maintenance energy requirement. I won't focus too much on some dietary manipulations that may modulate blood alkalosis, although there is a bit of research being done in that area at the moment. This is a relatively old textbook diagram, but it does show what happens in humans and dogs during heat stress. So on the left-hand side here, during a cool environment, you can see blood flow to the internal organs is great in both the human and in the dog. Under hot conditions, though, you can see that there's a reduction in blood flow to the internal organs and an increase in blood flow to the periphery. And you'll see in the dog that there's an increase in panting, trying to increase loss of heat from the lungs. And if you look at this example here, this is in humans, and this is blood flow to the skin. You can see that early on, when there's no heating, blood flow here is about 5 mils per 100 mil of tissue. But then once we start to heat the human, you can see a dramatic threefold increase in blood flow to the skin, which is maintained while the heat treatment is maintained. Then when we reduce temperature, we revert back to where we were beforehand. So, you know, a threefold increase in blood flow to the periphery, to the skin, in an effort to dissipate heat from the body. So this occurs in humans. It also occurs in our domestic species. And you can see visually here what's happening in the pig. So the pig on the left-hand side here is under thermoneutral conditions. If you look at during heat stress, so this is probably about 35 degrees, you can see that the skin is a lot redder as blood is being diverted to the skin, And you can see that that pig there is open mouth panting and could be in some distress. And so what we see typically is these sorts of responses. Now, I'll just take a little while to talk through this slide because it's very similar to some of the other data slides that I'll show during this presentation. So on the bottom axis, we'll have time of the day, and on the y-axis, we'll have whatever variable we're measuring. And what we typically do in our experimental regime is that we'll increase the temperature starting at about 9 o'clock in the morning, and we'll go through to somewhere between 3 and 5 o'clock. And then we'll reduce the temperature down to around 26 to 28 degrees overnight. We won't take the temperature all the way back to 20 degrees because we're trying to simulate what happens during a heat wave, where the temperature increases during the day, at its highest in the early to mid-afternoon, and then declines overnight, but doesn't decline back to thermoneutral conditions. And what you would see in virtually every domestic species that we've worked with, if we look on this axis here on the left-hand side, we've got rectal temperature. In the thermoneutral pigs, you can see a slight increase in rectal temperature over the day, and this is because the animals eat, so they generate endogenous heat being produced. If you look at those animals that are subjected to heat stress, they start off higher because the temperature has not come all the way down to 20 degrees, but there's a marked increase in rectal temperature over the first few hours of the application of the heat, reaching a plateau, and sometimes it drops away a bit. And then once we remove the heat, then the temperature will decline. So, somewhere like an almost 2-degree increase in rectal temperature in these pigs when the temperature was increased from 20 to 35 degrees. On the other hand, you can see respiration rate. So under thermoneutral conditions, these pigs are sitting at about 25 to 30 breaths per minute. Those animals that have been exposed to heat are already at about 55 breaths per minute, and then a dramatic increase up to around 200 breaths per minute. So more than 3 breaths per second. And I would challenge anyone in the audience to try to keep this sort of panting rate up for anywhere more than a minute. You may even be battling to get to a minute. And this is particularly important for the pig because unlike the popular adage, pigs actually do not sweat. They do not have functional sweat glands. They don't sweat. The only way they can get rid of heat is through either increasing their respiration rate and panting, or through loss through the skin, through either radiant heat loss from the skin or wallowing, as they do in the wild, or having drip coolers, dripping on top of the pig. So the pig is actually particularly susceptible because of this lack of functional sweat glands. So when someone says sweat like a pig, what they actually should be saying is sweat like a human. And so that was rectal temperature, but this is some work that we did a few years ago, where we were looking at using these capsules that we can place in the various regions of the gastrointestinal tract to measure gas production as well as measure temperature. And I'll just take you down to the bottom right-hand corner here, where we're looking at stomach temperature here in animals that are exposed to 35 degrees temperature. You can see that stomach temperature gets up to around 42 degrees. So rectal temperature might be 40 degrees, but in fact, internally, within the gastrointestinal tract, the temperature could be up to 42 degrees. So quite dramatic changes and fluctuations in what we would really refer to as core body temperature there. And if we just look at this diagram here, this is from Nick Garbler's laboratory in Iowa State University. I think it's quite a dramatic and clear depiction of what happens to the gut during heat stress. So many people will talk about heat stress as being a disease of the gut. So if we look at this diagram up here at 0 hours, so this is a cross-section of the small intestine. So inside here is the lumen of the small intestine, and these finger-like projections are the villi. Now, these villi are very important for absorption and digestion absorption of nutrients. They increase the surface area of the small intestine, thereby increasing the area for digestion and absorption. Then down between the villi are what we call crypt, and this is where the tight junctions are that prohibit the influx of foreign matter, such as endotoxins that might be produced by some of the bacteria in the gut. So at 0 hours, sitting at about 20 degrees, you can see quite healthy villi, elongated, and you can see that these tight junctions are very tight. But just 2 hours after increasing the ambient temperature to 40 degrees in this case, you can see that these villi are misshapen, they're sloughed off, they're shorter, and you can see starting to open up these tight junctions, which are becoming no longer tight. By 4 hours, even further damage to the villi. You can see some sloughing off of tissue here, and probably appearance of bloody mucus in the feces. And by 6 hours, it looks like a complete disaster, where these villi have been severely damaged, and the tight junctions are certainly no longer tight. And this is where we would get invasion of endotoxins. And so, as I said, a lot of people talk about heat stress as being a disease of the gut, and this is because of the diversion of blood away from the gastrointestinal tract. So no longer is oxygen being taken and nutrients being taken to the gastrointestinal tract, and no longer are end products of metabolism being taken away. So there's a build-up of reactive oxygen species which are causing oxidative stress. And just, I suppose, a reminder to us that this would also occur in humans. And one of the analogous comparisons, I suppose, to this is a condition called runner's gut, where long-distance runners can suffer from something very similar, as the blood is diverted away from the gastrointestinal tract towards the muscles and the periphery, both to supply energy for the running activity, but also to dissipate heat. We can see some classic examples of runner's gut and bloody diarrhea in runners during long distance races. It's not quite the same, but it is somewhat analogous. Okay, I'll just step back. This is a different species. We're now talking about dairy cows, and I put this in just to remind us that the nutritional strategies that I'll talk about today do not replace good management. They're an adjunct to good management. And by good management, I mean things like supplying sufficient water, supplying shade, and within the dairy or within pig sheds, to ensure that there is adequate ventilation, and where appropriate, some sort of fan or mist cooling or drip cooling. So here you see some dairy cows, some nice pasture there, but they're not just going out in the pasture. They want to seek out shade. Here's an example of an individual cow, cow 1280 on the 24th of December 2018, out grazing at pasture. About 10 days later, when we had a heat event. This is at our Dookie farm up in northeastern Victoria. Here's the same cow seeking shade, not interested in grazing at pasture, and you can see open mouth panting and salivation. So important to provide things like shade. Then here you see some dairy cows that are actually seeking water, not just to drink, but to cool down. Okay, so that's an introduction to heat stress. I'll now talk a little bit about oxidative stress and heat stress. I've already sort of introduced the concept that oxidative stress occurs in the gut. So what am I talking about there? Well, what I'm talking about is the build-up of oxidative species and free radicals. And this production of free radicals, this production of these reactive oxygen species, is a normal process in functioning and metabolism. And so, you know, we expect the production of these antioxidants, and under normal conditions, the activity of a variety of different enzymes, oxidases, cyclooxygenases, lipogenases, peroxidases, and dehydrogenases, will actually rid the body of an accumulation of these reactive oxygen species. So what we see is that, in fact, we have a balance. But oxidative stress occurs when there's an imbalance between oxidants and antioxidants, which can be due to either excessive production of free radicals, these reactive oxygen species, or it could be because there's a deficiency in the antioxidants that we provide to the animal. So it's ultimately a failure of the antioxidant system of the animal. And this is why we've focused a lot of our research on antioxidants, dietary antioxidants, and ways in which we can improve the antioxidant status of animals when we're trying to combat heat stress.
Thank you very much, Professor Danchet, for the initial part of this presentation. Is absolutely interesting and insightful. Before going into the antioxidant strategy, I would like to publish the second of the 4th poll for today. And I would kindly ask each of you to give the reply right now, and then later on waiting for the answer coming from the presentation of Professor Danchet. I can see some answers are already coming. Thank you very much to all the attendees for participating. One more minute, and then we can proceed. Please don't forget to choose 3 of the choices. Okay, thank you very much. The poll will remain open, so if you feel like answering the poll, even during the presentation after Professor Danchet will already share some of the insight and answer, feel free to do it. Professor Danchet, thank you very much. The stage is yours again.
Okay, thank you very much. So I'll now go on and talk about antioxidants. And when I talk about antioxidants, the principal ones that we've done our research with have been vitamin E and selenium. Now, these are not the only antioxidants that exist, but they are the ones that we've focused on our research. And we will obviously be looking at others in the future, including plant-derived antioxidants. But why have we chosen vitamin E? Well, it's the major lipid-soluble chain-breaking antioxidant in cellular membranes. It protects against lipid peroxidation and maintenance of membrane integrity. And it's also involved in a range of immunomodulation and gene regulation. Selenium, well, it is the structural component of a number of different enzyme systems, including the important glutathione peroxidase enzyme, which is involved in removing oxidants. It's incorporated in the proteins as selenoproteins. So there's a range of different proteins that are important for protecting against oxidative damage. And it's fully ionized at physiological pH, meaning that it's actually important for redox reactions. Okay, so this is a Mangalitsa pig. It's just to remind you that some of the data I present will be in pigs, and some of it will be in sheep. So this is my model cross between sheep and pigs that I like to show. So some of the data I'll present in the next little while will be data generated by Surinder Chowan during his PhD. And he did a lot of his work with sheep, but in fact, it's equally applicable to dairy cows. So this is some work where Surinder fed a control diet. And when I say a control diet, I'm talking about a diet that contained what we consider to be the normal levels of vitamin E and selenium for maintenance. Okay, so it's not deficient. These diets were not deficient in selenium or vitamin E. They were fed at the requirements, at the NRC or whichever requirements we're using, depending on which species. So be aware of that. I'm not talking about animals that are deficient when I'm talking about the controls. I'm talking about animals that are fed at their requirements. Then there was animals that were fed that control diet and exposed to heat. And then there were animals that were fed with what we call super nutritional levels of selenium and vitamin E. So they were between 5 and 10 times the levels that we would normally feed. And then animals that were fed that high antioxidant diet and also exposed to heat. Now, what we see under thermoneutral conditions is, in fact, over the day, there's a small increase in respiration rate that declines after the heat event. That declines after, say, 5 o'clock. Because these animals have been fed once a day, there's a small decline as that accumulated heat from the endogenous production in response to feed consumption goes down. In those animals that are exposed to the heat stress, so initially there's a higher respiration rate because remember I said in our model we don't bring the temperature back down to thermoneutral, we keep it slightly elevated to simulate what happens during a heat stress event. In both dietary treatments, there's an increase in respiration rate, again up to around 200 breaths per minute. But you can see that those animals that were fed the antioxidant diet, so the diet high in both selenium and vitamin E, there's a lower increase in that respiration rate. Similarly, if you look at rectal temperature, we see the same sort of pattern, but there's a lower rectal temperature increase in those animals that were fed the high selenium and high vitamin E diet. Now, interestingly, if you look at the control animals that were exposed to heat, you can see that there was about a 14% reduction in feed intake. So it's a normal adaptation to heat stress, is the animal tries to reduce endogenous heat production by reducing the amount of food that they eat. If you look at the animals that were fed the high selenium and high vitamin E diet, though, they maintained their feed intake. Okay, so not only did they maintain their feed intake, they actually had lower rectal temperature and lower respiration rate. Now, you would expect that rectal temperature and respiration rate would go up because these animals are generating more heat. So in fact, the combined effect is that they increase their feed intake and they maintain their feed intake and they have a lower respiration rate and lower rectal temperature, indicating a partial protection against heat stress. And if you look at some of the genes that are involved in the antioxidant system, you can see this is glutathione peroxidase over here. You can see during heat stress there's a decrease in the control animals. If you look at the high vitamin E, high selenium diet, we're able to stimulate that enzyme. It was reduced during heat stress as well, but it was maintained at about the same level as it was in the control animals. If we look at SOD2, this is another enzyme involved in antioxidant system. You can see a similar response. There's a decrease with heat stress. There's an increase with the selenium and vitamin E diet, such that under heat stress, the levels in the selenium and vitamin E fed animals were similar to those that were in the control diets under thermoneutral conditions. So we've seen some physiological responses, we've seen some feed intake responses, and also demonstrated that that some of the enzyme systems that are involved in protecting against heat stress and the oxidative damage thereby caused are elevated as well. Another experiment that was conducted by Surinder, so this one was to look at either low selenium and low vitamin E. So again, when I say that, I'm talking about those animals that were fed at requirements, or those that were fed a high selenium, low vitamin E diet, those that were fed a low selenium, high vitamin E diet, or those that were fed a combination of both, thermoneutral and heat stress. And this is actually measuring hydrogen peroxide in the exhaled breath. So these animals increase their panting during heat stress, and this is measuring the amount of hydrogen peroxide that's in that exhaled breath. You can see in the animals that are fed the control selenium and vitamin E diets, there's an increase during heat stress. But then you see that no matter which antioxidant we use, whether it was selenium on its own or vitamin E on its own or combined, we're being able to show this protection and reduction in exhaled breath condensate. So both vitamin E and selenium, although they act differently, are seeming to at least have similar effects here on hydrogen peroxide production. This is another study conducted by Surinder, and this one was looking, this is in lambs, and this is looking at either a control diet, a moderate supplementation, or the high level of supplementation of both vitamin E and selenium, and looking at some of the systems involved in protecting against antioxidants. So this first slide over here, this first graph here is the level of reacting oxygen metabolites. And you can see during heat stress that there's, in the control diet, there's an increase in these reactive oxygen species. But as you increase the level of vitamin E and selenium, you can see that the increase is nowhere near as severe. And then if you look at advanced oxidation, so these are damaged proteins, this advanced oxidation protein products. So these are damaged proteins that have been damaged through oxidative stress. You see an increase during heat stress on the control diet, and again, as the increase in selenium and vitamin E occurs, there's a reduction back to those control values. Okay, so I'm just going to visually show you what we're talking about. So, oops, that didn't work. Okay, so here is this sheep is being fed the selenium and high selenium and high vitamin E diet, and it's at heat stress conditions. There's some rapid panting, but here is its control counterpart, and you can see much more visible signs of heat stress. So here are two sheep side by side together, but this control animal is showing open mouth panting as it tries to dissipate heat. So I think a quite clear articulation of the sort of effects that we're talking about here. So just to remind you again about what happens in the gut, I showed this earlier. This is what happens during heat stress. So we've been particularly interested in what happens to the gut when we feed these high levels of selenium and vitamin E. So this is some work that was conducted during Fan Lu's PhD. This is in pigs. And what I'm showing here is transepithelial resistance, and we're looking at increasing doses of selenium and vitamin E. So once again, this selenium at 0.2 parts per million and vitamin E at 17 international units is at what we consider to be requirements for these pigs. Okay, Then we increase about 5-fold in selenium and about 10-fold in the dose of vitamin E that we provide. Excuse me. And what we're looking at here is transepithelial resistance in the small intestine. So we've slaughtered these pigs and we've taken samples of the small intestine, and we've measured the electrical resistance across that small intestine piece, that segment. And what that measures is if there's a decrease in that transepithelial resistance, that means that the tissue is likely to be more permeable to extraneous products, such as endotoxins. So it's less protective. So what you see is that in this control diet, yes, when we apply heat stress, there's a reduction in transepithelial resistance. But as we increase the level of vitamin E and selenium, we see that at about the selenium fed at about 0.5 and 100 of vitamin E, we're back around where we were with the control animals. Okay, so not only is selenium and vitamin E protecting with respect to feed intake in the sheep and respiration rate and rectal temperature in this model in the pigs, it's also showing that we've actually reversed the effects of heat stress on gut integrity. This is showing something similar. This time we're looking at 4 kilodalton dextran permeability. So this is measuring the permeability of the gut to dextran. And if you see an increase in this permeability, it's showing that the gut is increased in permeability to molecules of this size, of a 4 kilodalton size. And as we, I suppose the converse of what we saw with the other slide is, as there's an increase in heat stress on the control diet, you see that there's an increase in permeability. But as we increase the level of selenium and vitamin E in the diet, we're back down around where we were with the control animals at about 0.5 and 100 dosage of the selenium and vitamin E. And glutathione peroxidase, so this is in the same study, glutathione peroxidase, which is one of the key enzymes found in the gastrointestinal tract involved in protecting against oxidative damage, reduced during heat stress, and you can see a linear increase as we increase the levels of the antioxidants in the diet. And if you look at the ratio of GSSG and GSH, so this is an indicator of the reduced state of the enzymes, increase with heat stress, and declines with increasing dose of selenium and vitamin E, again indicating protection with these increasing doses of antioxidants. Okay, so that's a summary around antioxidants. I've focused on selenium and vitamin E, but in fact, we are searching still for other alternatives, and we're looking at plant-derived molecules that may actually provide antioxidant protection, because we're very keen to present a suite of different compounds. I'll now move on and talk about chromium. So why am I talking about chromium? Well, one of the reasons is that chromium acts as an insulin mimic and actually can be beneficial during diabetes. So why are we interested in diabetes? Well, this slide here is in humans, and it's looking at the ability of a normal human or a type 1 diabetic human to dissipate heat during exercise-induced heat loads. So if you look at heat dissipation as the amount of exercise increases from 400 up to 600 watts, you can see that normal heat loss in an individual increases. Okay, there is an increase in rectal temperature, but it's not severe. On the other hand, the type 2 diabetic individual who has an inability to partition blood to the periphery is not able to get rid of heat as well. And so they reach a plateau in their ability to dissipate heat and consequently see a dramatic increase in rectal temperature. So for individuals who are diabetic, heat stress presents a real problem. So why is that important for livestock? Well, during heat stress, animals may actually decrease insulin secretion and increase fat deposition. And we know that blood flow to the skin is reduced when insulin is low or when animals are insulin resistant or diabetic. And chromium acts as an insulin mimic and may actually increase blood flow to the skin to help lose heat during heat stress. So that is why we've chosen to investigate the role of chromium in heat stress. And just to demonstrate that we do see these changes in insulin metabolism, we've just published a paper here where we looked at nanochromium picolate, and we found that it actually improves the gene expression associated with insulin signaling in both porcine skeletal muscle and adipose tissue. So we've been quite clearly able to demonstrate effects at a gene level. But what do we see in the live animal? So similar design here. This again is work from Fan Lu's PhD, looking at rectal temperature and respiration rate. And we see the typical response during heat stress. But what we do see is that those animals that were fed chromium at around 400 parts per billion had a lower rectal temperature, and in particular had a markedly lower respiration rate. So yes, chromium does seem to be having some beneficial effects there. If we look at what happens in sheep, here are some sheep that were fed dietrychromium picolinate as well. During heat stress, there was a reduction in feed intake, but in the animals that were fed the nanochromium picolinate, there was nowhere near the same sort of reduction in feed intake. This is work that's in press from Alex Hung's PhD. And if you look at what happens to average daily gain during heat stress, there's a dramatic reduction in average daily gain in those animals fed the control diet. Those animals fed the nanochromium picolinate. Yes, there is a reduction in average daily gain, but it is nowhere near as great as what you see in those animals that were fed the control diet. So I think quite clear demonstration that there's also a role for chromium in improving the animal's ability to handle heat stress. Betaine. So the reason we're looking at betaine, it's actually an osmolyte. It improves tissue hydration and tissue fluid balance, reduces energy expenditure and maintains iron balance, can also act as a methyl donor and antioxidant. And why are we interested in it? Well, during heat stress, we get hyperosmotic stress, but what happens is we get a compensation with iron pumps. So to maintain water balance, we have these iron pumping within the cell. So we can maintain cell volume, excuse me, but that comes at a high energy cost. What happens when we feed dietary betaine is that betaine actually gets into cells, acts as osmolyte, and reduces the need for this iron pumping. And as a consequence, there's a lower energy cost. So you actually get a reduction of maintenance energy requirements of between 5 and 10%. And what happens when we feed betaine to animals? So this works from Hugh, just been published, looking at respiration rate in control animals, those fed betaine. You can see that dietary betaine reduces respiration rate. If we look at the gut, we can see that this works out of Nick Gabler's lab again in Iowa State. We can see that in the ileum, cecum, and colon, during heat stress, there's a reduction in transepithelial resistance, which is indicating that the gut is becoming leaky and permeable to extraneous compounds. But if you provide a dietary betaine during heat stress, you can reverse this effect on the gut. If you look at what happens in dairy cows, so this is some work that we did during summer up at Dookie about 2 or 3 years ago. Milk yield was increased by about 1.4 kilograms per day. Milk protein was increased. Milk fat was increased. Concentrate intake was increased. So these animals are grazing at pasture, but we provided concentrate in the milker. Milking frequency was increased, and rumination time was increased. So quite clear indications that this protected against betaine protected against heat stress during normal summer conditions. But in particular, where we saw the greatest responses in milk yield was when there was a very high overnight temperature here. So if you look at the graph down the bottom, we're looking at the effect on milk yield. You can see that when there was hot overnight conditions, milk yield dropped away in the control animals, came back, dropped away again, whereas there was much less of an effect in those animals that were fed the dietary betaine. Okay, I'd just like to wrap up now with the last little bit on grain treatment. And the reason we're interested there is that different grains actually ferment at different rates, and they can have an effect on heat stress as well. So this is some work done with dairy cows at Dookie, experiment conducted by Peter Mote, where we used infrared cameras to look at flank temperature, and in particular looked at the difference between the left and the right flank, because where the rumen is located on the left is a clear indication of fermentation in the rumen. And what we saw in this was, in fact, those dairy cows that were fed a diet containing wheat as the source of starch had higher temperatures than those that were fed corn. They had a higher left flank, they had a higher right flank, and they actually had a higher difference between the two, 1.1 degree versus 0.7 degrees daily. And then if you look at postprandially, 1.4 degree difference versus 0.7. So quite clear differences between corn and wheat in the temperature of the rumen. So what does that mean for heat stress? Well, the reason why we see this is, in fact, maize or corn ferments at a much lower rate than wheat, ultimately, you end up with the same gas. This is an in vitro measure of fermentation, but the rate is slower in maize. Now, if this occurs in the animal, then you would expect less heat being produced in the rumen as well. And some work done by Paula Gonzalez during her PhD, looking at thermoneutral conditions here, looking at heat stress, and there's the animals fed 1.3 maintenance and those fed 1.5 maintenance. Looking at respiration here, you can see that those animals that were fed the corn diet had a lower heat production or respiration rate, than those animals that were fed on the wheat diet. So indicating quite clear differences in the rate of fermentation of these diets and their impact on ability to handle heat stress. And rectal temperature, you see a similar pattern, with wheat being higher than corn. And if you look at the differences between the left and right flank temperature, again, quite clear differences between the corn and the wheat diets and their ability to handle heat stress. We've also gone on and looked at sodium hydroxide treatment of wheat to try to slow down the rate of fermentation of wheat, because corn is not always available. And what we see is, when we've done our in vitro fermentation here, we've seen that the sodium hydroxide treated wheat has a similar fermentation pattern to corn. And if we look at what happens in the animal, this time we fed them at 1.8 maintenance under thermoneutral and heat stress conditions, and then at twice maintenance, you can see that that the sodium hydroxide treated wheat is somewhat intermediate between that of those fed corn. In fact, one of the reasons we don't see such a greater effect under ad lib conditions is, in fact, the animals eat more of the sodium hydroxide treated wheat. But again, quite clear indications that the rate of fermentation of grain in a diet can also have an impact on heat stress. And we're currently looking at other means of slowing down the rate of fermentation of wheat, because people don't really want to work with sodium hydroxide in their systems. So finally, to conclude, I think it's quite clear that heat stress will increase in severity and frequency, and that will present problems for humans and the livestock health and productivity. I think I've shown quite clearly that heat stress can have severe impacts on gut integrity, but these can be partially ameliorated by antioxidants, betaine, and possibly some other plant-derived compounds such as polyphenols. Heat stress increases respiration and rectal temperature, and again, these can be partly reversed by some of these compounds. I think there's a role for chromium improving blood flow to the skin, and also that slowing down fermentation of grain can also protect against heat stress in ruminants. And just to sort of demonstrate that this is the result of an enormous team over the years, probably work that's been conducted over the last almost 15 years. And this is, well, the latest iteration of the students and colleagues who are involved in heat stress. There are many others who have come and gone as well. So thank you very much for listening to me.
Professor Danchet, thank you very much for the very insightful presentation. It's been very, very interesting to see what is not only the severity and the frequency of the heat stress, but as well the huge impact that it does have on the animals, on many organs. Gut is absolutely at the priority of the point that we need to consider to help through the nutritional strategy to take care. I would like to call on stage Pietro for the second presentation. And before that, I've just published the third of the fourth poll. I would like all of you to take 1 minute to answer this poll. I just want to share that there are already few questions popping up. I'm really happy for that. I can't wait to have the Q&A session later on because there are a few questions that are really interesting. Okay, the poll is available. Please do not forget to answer and to pick the 3 top factors according to what you think that may impact feed intake. Okay, so while the poll is on, I would like to introduce Pietro. Pietro Celli is Adisseo Asia Pacific Regional Category Manager for Ruminants and for Palatability. In his previous career, Pietro joined the University of Sydney as a senior lecturer in the ruminant health and production, focusing in study into animal physiology, nutrition, and reproduction. Today, he will inspire us on the importance of maintaining redox balance and feed intake during heat stress. I think the poll is almost ready. Okay, I think we can proceed. Pietro, the stage is yours whenever you're ready.
Thank you, Ricardo, and good morning, everyone. So I have few minutes, about 20 minutes, to entertain you with few slides, and I'd like to address 2 topics. One on the importance of the maintenance of feed intake, and the other one following very much what Frank has presented, the maintenance of redox balance. So I've got a couple of examples that I'd like to go through with you, and that's the reason why the first poll very much focus on feed intake. So the first part of my presentation will be on feed intake and the importance of maintaining feed intake with the example of the high prolific sow. As you probably can see there, we've got a lactating sow with about, I think I counted 11 piglets. counted the number of legs divided by 4, so I'm not sure whether how accurate is that, but quite often we see more than a higher number, and that what it means is that the nutritional requirements for the sow are also increased. And essentially the key here, the take-home message here for the modern prolific sow, everything must be done to maximize lactation feed intake. So this particular window is crucial for the health and the productivity and longevity of the sow. But not only that, as I'll show you, is very important also to maximize and to boost piglets performance. And with this slide, which is quite busy, there's a lot of information there. So I would like you to focus your attention on these two lines here. So we got different stages on the productive life of the in the swine production cycle, starting from gestation to lactation, weaning, grower, and finisher phase. And the lines represent the feed intake. At the top, we have the target feed intake. At the bottom, the actual feed intake. So quite often we see at different stages of the cycle that feed intake actually does not reach the target that, for which the animal is being designed. So we essentially don't allow the animal to achieve its performances, its genetic potential. And crucial windows are around lactation in particular, but also other stages. I think we're all very familiar with the challenges in piglets, especially post-weaning. There's could be a quite severe dip in feed intake. So what I'm trying now to introduce is the concept of palatability and palatability solutions that can allow us to bridge the gap, to make sure that actually we maximize feed intake to the potential. So try not to overachieve, go over that targeted feed intake, but to make sure that the animal can eat what the requirements are. And just very quickly, there's a lot of text in this slide, but just like to spend a few more seconds to align the fact that stress is a factor that can influence feed intake. And I think Frank has given us a number of examples in both swine and ruminants. And therefore, the presence of hot and humid and tropical conditions quite often represent a challenge for the animal in terms of feed intake. So we see a depression in feed intake in this situation, especially during the lactation stage. So what I would like to talk to you about is now going into some of the solutions and the application of this solution in the swine industry. In particular, the importance of sow feed intake during lactation and the impact on the performance and profitability. And this is a study that I'd like to share with you that is done in collaboration with Professor Silva in Brazil in tropical humid climate. Very briefly, very quickly, the experimental protocol. We had almost 300 sows divided in 3 groups with 1 control group and 2 groups receiving Crave, this product that stimulates, boosts feed intake at 2 levels, 250 and 500 grams per ton as an inclusion rate. So we had 3 replicates. These are the details in terms of sow parity. About 2.5, lactation period duration about 23 days, and no crib feed was offered during the lactation period. This table summarizes the results of the effect of Crave addition to the diets in lactating sows during heat stress condition during these hot and tropical humid conditions. And I'm just highlighting here the effect on feed intake. As you can see, a control was sitting around 5 kilograms per day, and a nice response from the 2 groups in Crave that received Crave in the diet. If we look at then the piglets, we see there's also an improvement in number of piglets that were weaned, an improvement in the average weight gain of these piglets, and an improvement in the litter weight at weaning. And last but not least, quite a nice response in milk production. Okay, at the bottom here, just for your curiosity, you can see the conditions that this experiment was conducted. So in summary, we see about almost a 30% increase in feed intake with Crave. More piglets that are weaned. These piglets are heavier, and this is the results of the fact that by achieving the target feed intake, the sow can increase the milk production. This milk is all the benefits goes to the piglets, and we see the improvement in the weaning weights that I show you in this table. Also importantly, there's no weight loss during lactation for the sow. So that's essentially a classic example of nutrient partitioning towards the mammary gland. So all that feed goes to the mammary gland. Something that also we looked at in this trial, I should say Professor Silva looked in this trial, is the distribution of weight in the litter. Okay, so looking at the control group, the 250 grams per ton and the 500 grams per ton of Crave group, looking at the distribution of the litter weights, and that's what we see. Essentially, more than 90% of the piglets in the 500 grams group for Crave were above 6 kilograms. So essentially, the take-home message here, we have an improvement in litter uniformity. Not a bad result here. Then I think a question that came up was, okay, what the palatability? What are the factors that influence flavor? And looking at the relationship here between the sow and the piglets, we know that we can influence flavor transmission before the piglets are born, and this is because some of the flavors are ingested by the sow, and some of these compounds maybe can go through the placenta, and the fetuses are exposed to these compounds, to these flavors. So that happens before the farrowing. After postnatal, what happens? Once again, because this flavor in the diet, the piglets, they see what the sow is eating. The piglets can smell and taste these flavors, and some of these flavors can also be transferred through the milk. So that's the basic concept. And going back to this, the life cycle for swine, we decided to, okay, let's target this window here. So the sow nutrition during lactation and the creep starter feed for the piglets. Can we actually link these two stages together with our solution? And something we came up with is, so we have the Crave flavor with Crave AP that I presented just some data in these heat stress conditions in Brazil, and this Deli Start K-Link, which essentially has three components. This is OptiSweet. We have the Crave flavor that also the sow are eating, and another appetite stimulant is more for the umami flavors, targets more the protein in the diet. With this concept in mind, we once again, this is a study originates from Bruno Silva lab in Brazil, and looks at the effect of the maternal nutrition, so feeding Crave in sows, and once again similar protocol in the sense that we had two groups of sows, 90 each, a lactation period of 23 days, and looking at the performance of the sows during this period, during lactation stage, during summertime, so heat stress conditions. I like to draw your attention to feed intake first. So Crave, did the Crave work? Yes. Once again, we see an improvement in feed intake. We can boost feed intake under heat stress conditions. What happens to the piglet? Is there improvement in piglet performance? Yes. we see there's a 10% improvement in average weight head weaning, and in average gain in the ADGs. There's an improvement in there. And do we see a response in milk production? Yes. So consistently, we see this in this trial compared to the previous one and in many others that have been conducted with Crave. So first question, does Crave work in sows? Yes. So we have this answer there. Now, what happened to the piglets? So essentially now I'm showing you a 2x2 factorial design experiment whereby the progeny from the sows were received after farrowing. So already this crib feed was offered, either a diet with or without the kaolin, the daily start kaolin in the diet. Okay, and same kind of approach was done for the piglets born from sows that received Crave. Some had a control feed, and another group had the kaolin, the daily start kaolin. So comparing now the performances of these or the progeny of these piglets, and now focusing on the first 7 days, we see an improvement. Okay, the question: what happens if we do nothing with the sow and we just give the daily start in the piglets? Do we see an effect? Yes. We see an improvement in feed intake and also a slight improvement in daily gains, about 6 and 7% respectively. What happens if we compare group 1, so our control group, with the piglets born from sows that were fed Crave and received a control diet? We still see, so that's the maternal effect. Piglets born from sows that that received Crave in the diet perform better. So there's a maternal effect there, quite a nice one. But then, once we look at compare the control group with group 4, where the piglets born from sows that were fed Crave and also had the daily start in the diet, these numbers jump quite up. 30% increase in feed intake and 40% increase in daily gains. So quite a nice response. And last comparison here is the comparison in relation to group 3 and 4. So essentially, group 4 outperformed also group 3 in terms of feed intake and daily gains. So very nice results. When we look at piglet performance post-weaning, and this is data pertinent to the first 7 days after weaning, once again, we have in this table the 4 groups with the 4 different treatments. Group 1 being the control group, and group 4 is the piglets that received the piglets and the sows, the piglets born from sows that received Crave and daily start post a birth. Once again, excuse me, we see a nice effect in terms of daily gain, average gain, and in feed intake. So, that also highlights how this solution can minimize the negative impact of weaning on feed intake. So to conclude this first part of my presentation, I'd like just to summarize this concept of sensory imprinting and the benefits that we can have with this program, with the Crave and Daily Start. So we can see benefits in the sow in terms of increased feed intake, increase in milk production, better condition at weaning, and therefore we see also improvement in terms of fertility, shorter weaning to estrus intervals, but also we're seeing with all trials in the field improvement in longevity. Not a bad outcome considering now the recovery from the impact of ASF in several parts of this region. When we look at the piglets, we see that we can have more piglets at weaning. These piglets are heavier, and also the litter are more uniform. Another nice outcome. These solutions can also, once again, optimize feed intake and therefore reduce problems also at weaning. And with that, it comes a number of benefits in terms of daily weight gains and therefore shorter time to finish. With this, I'm moving on to the second part of my presentation. And then I do that, I like to publish the last poll for today, which should appear now on your screens. And we're going back to e-stress. So trying to see how all the information that has been presented to you so far from Frank's presentation. So I give you a few seconds to pick your three answers there for this poll. And in the meantime, as you do that, I just quickly refresh your memory, the importance of all the information that Frank has shared with us in relation to the maintenance of redox balance, or this equilibrium between antioxidant and reactive oxygen species, and that quite often we can see a disruption of this equilibrium, of this balance, for a number of conditions. And specifically, Frank highlighted the importance of heat stress. And what I would like to discuss with you now is the utilization, the adoption of salicylate, this hydroxyselenium methionine, during heat stress conditions. And this is an example of a published study in dairy cows with eight multiple dairy cows. These are the details of the production levels. We had two treatments, and with a group that was fed sodium selenite, and second group was fed supplemented with salicylate. And we had two factors: either we had thermoneutral conditions or heat stress conditions. And this is the experimental protocol. So we had 9-day acclimatization, essentially, then thermoneutral condition for 29 days, and then a heat stress challenge. And essentially, with this slide, what I'd like to show to you is that the experimental protocol worked. So the challenge did, as you can see here, we have a number of parameters. Then the values during thermoneutral condition and heat stress conditions, and this is showing you that heat stress was able to increase rectal temperature, increase respiratory rate. Frank very clearly showed the impact of and the relevance of these factors in his presentation. A severe drop in dry matter intake, in milk yield, and an increase in some markers of oxidative stress, in this case hydrogen peroxide and nitric oxide. So the model worked, and we see an essentially imbalance in redox balance. So there's more oxidative stress. What happened to now some of the responses when salicylate was added to the diet in comparison to sodium selenite? We see here on your left is the serum total selenium, and on the right of the screen is the selenium content in milk. We can see that salicylate was able to increase serum content. So we have a better selenium status, but also there's an enhancement of the transfer of selenium into the milk. This is an important factor, and I'll come back to this in the next few slides. Once again, looking at this balance and the importance of maintaining redox balance, and we see a number of parameters or biomarkers of oxidative stress, looking at total antioxidant capacity, hydrogen peroxide, nitric oxide, as I mentioned before, or glutathione peroxidase and MDA, another marker of lipid peroxidation. We see that salicylate outperformed sodium selenite for all these parameters. So we have a better redox balance with salicylate. What happened to performance? In relation to performance, we see that no effect on dry matter intake. during heat stress, which was depressed by heat stress. So the antioxidant supplementation was not able to reverse this impact, but was able to maintain or sustain, I should say, milk yield. So there was a trend, but a 9% improvement in milk yield in the Seliseo group. And this is important because, again, going back to the transfer of selenium into the milk and to the mammary gland, essentially what we see is that we have a reduced free radical damage within the mammary gland when we have Seliseo into the diet. And with this, I'd like to show very quickly this slide, where is a study done in New Zealand, looking with in a customer here at issues with somatic cell counts. Once again, a similar protocol. In this case, we seeing the effect of Seliseo supplementation in relation to the somatic cell counts, and there's a clear effect of Seliseo, of Seliseo on somatic cell counts. So that's this line here. Once again, as I say, in line with what I was mentioning before, improvement in mammary gland health. So to finish off my last 2 slides, concluding this part of the presentation, we see selenium selenoproteins play a pivotal role in the antioxidant system and maintaining this balance. And the great advantage of Seliseo is that essentially he builds a sort of a selenium insurance policy, because you can build the reserves in the body, and therefore when antioxidants are needed, when selenium is needed during stress, the organism can draw on these reserves and then counteract the negative impact of auxiliary stress. Therefore, whenever there are challenges, whether they are during the transition period or in young calf or at mammary gland level, or during heat stress, the organism can draw on these reserves built up with Seliseo. And this form of selenium, or Seliseo, as I should say that not all seleniums have been created equal. This provides the most effective product, is the most effective product on the market that can create selenium body reserves and enhance antioxidant protection. Now, to finish off and try to bring all this together, so bringing this wine life cycle and the redox balance, hopefully I was able to show that palatability and solutions to increase feed intake during stressful conditions, during challenges, are very important. And this is a cornerstone of feeding behavior, and we can take advantage of manipulating feeding behavior to improve productivity and profitability. The maintenance of redox balance during heat stress can, you see, we have seen the benefits in terms of health and production. Both myself, I think, and Frank has showed that some of the approaches that can be used. And what I like to reinforce is that essentially nutrition can only begin when feed is eaten. So we can have the best formulated diets, but then if the animals do not eat them, nutrition cannot happen. And that's once again where palatability can provide our solution to overcome the decrease in feed intake during challenges like heat stress. And therefore, as simple as that, animals that eat grow. So stay hungry, stay healthy. With that, I thank you for your attention. I'll leave you with this very short clip of an excellent example of storing behavior in cows. They are excellent in picking little part component of the diet in there if they got extremely sensitivity in terms of taste and smell. And with that, I conclude and hand it over back to Ricardo. Thank you.
Pietro, thank you very much. Thank you very much as well for the presentation and for highlighting the very importance of the redox and feed intake in the animals during heat stress, and not only. With this, we can eventually move to the last session of today's webinar, the Q&A. We have already some questions that are popping out. I'm really happy about it, and I will take the chance to read all of them to the two speakers. Okay, so the first question is for Frank. Frank, about selenium, related to your presentation. Did you study different selenium molecules? What can you tell us about the different form of selenium in terms of their efficacy?
Okay, that's an excellent question, and I should have been clearer in my presentation, I suppose. So the selenium that we've worked with has been it's been selenized yeast. So we've worked with a range of different commercial products that have been selenized yeast. I suppose my summary of what I feel, and in light of Pietro's presentation as well, it's important that the form of selenium is organic. So selenite and selenate are quite dangerous. They can be toxic at the levels that we were talking about in our inclusion rates. Whereas organic forms of selenium are much safer, and they get incorporated into body tissues much more quickly. So I think I'm not going to say one product versus another, but what I will say is that you must use an organic form of selenium if you're going to look at the super nutritional sort of levels we're looking at.
Thank you very much. Pietro, do you have anything to add to Prof. Danshay's answer?
No, I think what I'll maybe like to say exactly, just reiterate the fact that organic selenium is the way to go, and that the Seliceo, I think we have a lot of data. Didn't have the time to go through it today, but is amongst the organic seleniums is the most effective one.
Great, thank you so much. Another question for Professor Danchay about betaine. Milk improvement due to betaine. It was natural or synthetic one?
So we work with natural betaine. There are very few side-by-side comparisons, but my impression from what I've seen, the little data I've seen, is that natural betaine has about 30% higher efficacy than synthetic betaine. At the same dose.
Great, thank you so much. And again for you, Frank, during your presentation, you had the chance to introduce different strategy, different additives to help mitigate the heat stress. But from a practical point of view, if you have to give one or two priority, which one of these would you recommend?
Okay. Look, in our hands, selenium, chromium, and betaine have probably been the ones that are most efficacious. If you look at some of the big commercial piggeries in Australia, lactating sows are often fed all of them during summer: chromium, betaine, vitamin E, selenium. One of the things is, though, that in some jurisdictions in the world, you can't feed any more selenium than 0.3 parts per million. Australia is actually relatively unique in that we can feed higher levels of selenium, except for Queensland. I would recommend that where possible, as long as the cost is not prohibitive, that you actually perhaps use a combination of them. But I think it does depend upon what are the clinical signs of heat stress that you're trying to mitigate against. Is it in the gut, or is it feed intake? And I think let that dictate what you do. Betaine and selenium, they actually accumulate in tissues relatively quickly, so they are strategies that you could introduce as you, in response to having a Weather Bureau alert, 5 days out. Or you can feed them all the way through. Chromium, I'm not so sure about. I just don't think we have sufficient data on how long we need to feed chromium for.
Good. Thank you very much. Pietro, anything in particular that you feel you want to share coming from the study and the application of the Silicio in that particular case?
I think that Frank has touched a very good point in relation to preventing instead of just reacting. So if we get a weather alert or a heat wave coming, and not all infrastructures have the options of mitigate, so providing shade or cooling misters and other technologies, whether now we're talking about swine or ruminants. Boosting building the antioxidant reserves is important. One. The other one, though, is, as I said, we need to make sure that the animals keep eating during the stress. Yes, antioxidant can provide a nice solutions for this, but then we need to make sure that they eat. And I'd just like to maybe mention the fact that the palatability solutions, they work both in stress or in thermoneutral conditions. So stress is only one of the causes that we see a decrease in feed intake. But it's not just the only one. It's not the main one. Yes, we are focusing on stress today, and I think the effect on dry matter intake is well established. It was well accepted. The concept that all doesn't matter which good is now we put in the feed, we need to make sure that the animals, they eat it, and that's where the advantage of these solutions, they can increase feed intake or maintain the target feed intake during challenging times.
Yeah, totally agree. Thank you so much. And both answers are definitely matching the next one. Next question will be specifically for both of you in this case. And since you mentioned about prevention and not only reaction, during the summertime or in prevention of the onset of summertime in Australia in this case, when would you recommend to start using antioxidant? And among the different solutions, which one do you think are the most consistent and cost effective?
Okay, well, I think it's horses for horses or cows for cows. Where you have sustained heat events, I think perhaps inclusion in the diet right the way through summer is applicable. Where you're having intermittent heat events, though, I think it's important. You can also have these strategies in the diet, or if it allows, you can bring them in and out. But it's a much more complicated scenario to do that. So I think during the hot months it's a good strategy. But of course, you have to weigh these things up on what is the return, and the return is not just milk yield. The return is also animal health and subsequent performance as well. In my experience is that the returns on investment are generally there for these strategies, but that might not always be the case.
I maybe following on what Frank just said. I agree what you said, Frank. Just like also to emphasize the fact that you can let's go beyond that 1 liter, 2 liters of milk in the tank, or whatever parameter we are looking at. Let's look also at the benefits now with this intervention. And of course, I'm focusing particularly on the antioxidants. And the palatability solutions that can have, we can go beyond exactly the extra 50 grams in ADG that you can see in a piglet or in a lamb or in a feedlot. There are the benefits, thinking now, especially in these situations where now looking at the swine industry rebuilding the stock after ASF. the possibility of improving lifetime performance, longevity, the welfare aspects associated with that, less use of antibiotics at weaning. Healthy piglets don't require intervention. So there are many benefits that can be then put on the table there for customers. And we can play the environmental, the sustainability aspect, the reduction in antibiotic use, AMR, and so forth. So we can easily go beyond that, that extra liter of milk in the tank for dairy cows, or that extra X number of grams in improvement in ADG. Yes, they are very important. Don't get me, I'm not trying to dismiss them. Of course, that's the starting point. But on top of that, we can see many other benefits.
Thank you so much, both of you. I totally agree with you. Thinking in long term will help definitely to increase the ROI. We have time for one last question, and is a combination, is for both of you speakers as well. Is about the utilization of the antioxidant, if they are recommended only during the heat stress, and in particular for Frank, since during your presentation we were focusing the attention on both combining the management strategy and the nutritional strategy. If the farmers have the possibility to cool down the cows with sprinkler, fan, and other equipment, would you recommend as well to use antioxidant in the TMR?
Okay. Look, I sort of must reiterate that at no time does a nutritional strategy replace good management. So I think it's just an adjunct to good management. So if you have the ability to provide shade, to provide cooling, then certainly do so. Unfortunately, in much of Australia and New Zealand, we don't have that capacity. Because sometimes these engineering opportunities are just too expensive. But if you do, yes. Now it's then up to you to decide, and perhaps by measuring respiration rate or some other parameters, whether you think a dietary strategy is also applicable. Okay, what was the first part of that question again? Sorry.
Was about the antioxidant. If recommend them only during the summer heat stress, or even during other period of the or stage of the life of the animals.
Yeah. Now, okay, so if there's no oxidative stress, then you need to be actually relatively careful with feeding high levels of antioxidants, because very high levels of antioxidants in a situation where there's no oxidative stress can actually create a pro-oxidant situation. So I'm not recommending across the board, but remember that oxidative stress occurs during pregnancy, it occurs during lactation, as well as during disease challenge, as well as during heat stress. So there are actually other situations where oxidative stress is critical.
I agree. If I can add my 2 cents, I agree with what Frank just said, and just to add, we as also our animals, we produce reactive oxygen species every day. The issue, once again, I think, is whether that balance is disrupted, and there are many conditions in animal in our livestock that can result in a disruption. So there are, of course, we can have different strategies. Of course, implementation across the board, there are always pros and cons, and we need to look at does it make sense from an economic investment. So this is a business that we are running here. There are advantages, some technologies that can essentially build this kind of insurance policy and mitigate the impact. And once again, it's not just a solution that can prevent all problems. The good management needs to also be combined with that. So you need to have also an holistic approach, not just a feed additive that fixes all the problems. So keeping in mind that free radicals are produced every day, every hour, every time animals eat. Every time there's is inflammation, and the tricky part is quite often we see subclinical conditions that lead to acute inflammation, and therefore production of free radicals and disruption of the redox balance. That's why it is important then, according to what the issue that we're trying to address, to customize solutions that can actually fit that particular scenario.
That's great. Thank you very much, Pietro. Thank you very much, Frank. I would definitely take the chance to conclude today's webinar. First of all, let me thank again Frank and Pietro, the two speakers of today's webinar, for the insightful information that they share with us. I want to thank as well all the attendees for the participation and for the contribution. I would like to just to recap a couple of key take-home messages coming from the speakers. First of all, heat stress is coming more often and with much more severe impact in our animals. Nutritional strategies and management have to be combined, and alone, unfortunately, are not enough to counteract the negative impact that heat stress may have, in particular on the gut and the absorption of the nutrients. As we learn from Pietro as well, feed intake is a key aspect not to forget during the heat stress, as well as the redox balance. Before ending the session, I want to remind each of you participants that you will be receiving an email with the link to the webinar recording and the answer to the question that today has been discussed, and others that unfortunately we didn't have enough time to do. I want to thank you again, all of you, and I wish you all a very good day and a wonderful weekend ahead. Thank you so much. Thank you, speaker. Thank you, Frank. Thank you, Pietro. And goodbye to everyone.
Goodbye.
Okay. Goodbye.
Thank you. Thank you, Frank.
Cheers. Thanks a lot.